can't say Jarosiewicz.
Dr. J, I like that.
Dr. J, and Charles Liu, and Ted Berger. We need a fourth chair.
You know, maybe I can monitor here.
Good.
I think that'll work. Yeah.
That sounds great.
Is that okay? All right, great. Well, that was great, Kent. You know, as I mentioned last night, this is LA, you know. You say, let's do it. The answer is okay. Let's do it. Okay, so that was terrific. I think that when one hears 3 talks and then Kent sort of piling on to give an overall sort of view of things from DARPA's side, and since DARPA's really funding much of this, I think there's probably a lot of questions on people's minds. So I thought that it'd be a good idea to have what amounts to a roundtable discussion from many of the experts that have joined us in this conference. So actually, Dr. Anderson and I, at lunch, talked about some potential questions that might be of interest to the audience. So I thought what I'd do is ask each one of these questions in turn and give each one of the panelists an opportunity to sort of answer these questions and of course to take questions from the audience. So I suppose the first question would be that you know, I think that what we've seen here today is today and last night is a view of what the current state of the art is of brain-computer interface and what technology and advanced prosthetic technology is capable of. So I suppose the question that would be interesting would be, how would you individually see the future of this technology going forward? Dr. Berger, do you want to start with that?
Sure. Well, I think it's going to surprise people how fast it will move. I mean, the demonstrations that I've heard from my colleagues today, I mean, they're already helping real humans do real things. The kind of prostheses that we're working on, which have to do with memory and sort of higher thought processes, that may take a little longer, but the usual reaction is the kind that Kent was talking about, which is, you're crazy. How many times I've heard people say that we're crazy? I don't know if I could count them, but and no one would have predicted that we're as far along as we are today. So I think it's really going to move a lot faster than well, certainly much faster than the naysayer types, but it'll move much faster than people think. And there's always this barrier of touching the human brain, and I think that will melt. And I think that what really needs to happen is a merging of medical needs and these experimental needs, so to speak. And I think once some of the platforms, I don't know if I should really talk about humans as platforms, but nonetheless, this clinical experimental platforms, once they get developed, and we see how much can be accomplished, while at the same time helping meet the medical needs of these individuals, I think then it will just the barriers will crumble. So I think I would think in terms of less than 5 years that even the kinds of prostheses that I'm talking about will be in clinical trials. So,
okay. Dr. Anderson?
Well, I think in the short run, wireless will be important, and also the electrodes themselves, as you've heard, they tend to work from 1 to 3-4 years with degrading signals, so better, more robust electrodes. In the longer range, certainly the recording technologies, although they've been refined quite a bit, we're just really recording from a small handful of neurons. It's amazing. To me, it's always amazing that we can decode the amount we can from such a small population. So, microelectrodes, although they're a real workhorse of neurophysiology, it is very old technology. So, things may come along with molecular techniques and nano techniques for recording that will allow recordings from very large numbers of cells invasively. And this may come about if this brain mapping thing goes through, because I know some people are thinking of advancing the technology to be able to record from massive numbers of cells, single units. And then also non-invasive techniques that could, some advances perhaps in imaging that don't require strong magnets, for instance, may allow for non-invasive but very detailed spatiotemporal recordings.
Okay, Dr. J?
So John Donahue likes to say he's looking forward to the day when he'll be playing basketball with someone for an hour or so, and then he will turn to him, to John, and say, by the way, I'm using your brain-computer interface right now to control my whole body. So that's sort of where we hope the future will take us, but in the nearer term, we're collaborating with a group at Case Western, led by Bob Kirsch, who does functional electrical stimulation, and we're hoping to use our neural outputs to control the functional electrical stimulation device directly, so that we can reanimate the person's own limbs in the case of paralysis. And I think the wireless thing will be very huge for that too. If it's concealed within their skin, and then the functional electrical stimulation itself is also inside the skin, you won't be able to tell if a person is using it. It'll make it much more aesthetically acceptable than its current state. So that's our long-term goal. I think that pretty much captures my hope too.
Great. Dr. Berger, I think Dr. Anderson just brought up the sort of the brain mapping initiative, and I know that you and I have had some private conversations about that. Do you have any comments that you want to share with the audience about that?
Comments that I can make publicly. No, I think it's a great opportunity. We're still early in finding out what, I think, and shaping what that program might be like. But I hope that it's directed at more than just what it sounds like. I mean, mapping, knowing where something is in space, where every neuron is in space is important, I guess. But there's so much more that could be done with a program like that to understand not only where a neuron is in space, but what its functional properties are and how it transmits information to other neurons. I think is the kind of endpoint that would be the most helpful to this prosthetic effort. I mean, we need to know not just where a neuron is in 3D space, but who it talks to and what kind of language it uses to talk. The whole issue, what's really holding us back a lot, I think, is understanding the, quote, language of the brain, unquote. We really don't know what coding schemes are used by neurons and how information is represented in the codes of activity that cells exhibit. So a large-scale effort to understand not only where neurons are, but who they talk to and what kind of language they use to talk is going to be the most relevant, I think, to basic neuroscience, but particularly to our efforts to develop neural prostheses.
Dr. Anderson?
Yeah, well, it was pretty obvious with the genome, what the code was. And So you could, and a lot of that advance was in making cheap gene sequencing and high throughput. So it may be premature. Certainly, if you just collect a lot of data on brain activity like fMRI and anatomical connections and staining of different cell types, I mean, that's important stuff. It doesn't seem, you may end up with a lot of data that you still can't make a lot of sense of. But on the other hand, if it is mapping activity at a very, with new technologies, it may be that the technologies that will come out, and that's why I was emphasizing that aspect, that if new technologies come out, some really surprising things made. So, although they've made the analogy between the map of the genome and the brain, I don't think they're quite the same.
Great, Dr. J. Oh, the question was, there's recently in the New York Times there was the announcement of a large scale initiative to map the human brain from crevice to crevice, and so what I thought would be interesting for the audience to hear is your thoughts on what that would mean to efforts such as this.
Right. Well, I have some hope for the endeavor, but I'm also a little bit skeptical, I think. We've had basically the flatworm connectome for 20 years now, and we still have no idea what's going on with the nervous system of the flatworm. And it has, what, 200 neurons? So,
It's because the flatworm is deep in thought.
yeah, you can watch it thinking right there. So I'm sure it's going to be really helpful for helping to cure diseases that happen to afflict certain neuronal subtypes, for example, or things like that. But I'm not really sure that it's going to help us really understand the function of the brain any better than just sort of studying the anatomical samples sort of on local levels like we sort of have been doing already. And I'm not really sure how we could do that in a brain that's still inside of someone's head, which is kind of necessary in order to do neuroprosthetics with that brain. So those are my skeptical thoughts on the matter.
So that's terrific. Just to follow up on your last statement, the brain being in somebody's head and the limitations that presents for neuroprosthetics. Most of these, all these technologies that we've talked about today are based on invasive electrodes being implanted in specific regions of the brain. What are your thoughts in terms of non-invasive strategies in the short, mid, and potentially long term, as far as this is concerned?
So the nice thing, the reason that we decided to go with intracortical electrodes is because, as I showed in that one video, that the activity you can record is very specific. You can get individual spikes from individual neurons. Neighboring neurons in motor areas don't seem to necessarily do similar things to one another. So if you try to record with a system, for example, that's outside the skin, you're summing the activity of millions, possibly more, neurons and losing so much signal as it crosses through the skull and the skin. So it's never going to be possible, well, at least with technology that I can envision right now, to be able to get the sort of specificity that you can get when you go inside the brain with something that's completely non-invasive. It's possible with advances in fMRI or in MEG or EEG that we can get to a point where we can get much more specificity, but it's going to take a lot of work, I think, in the long term. For now, we're making pretty good progress with sort of non-intuitive mappings. If a person can imagine opening and closing their hand to move a cursor in one dimension and wiggling their foot to move it in another, that works pretty well. But it takes a little bit more learning on the part of the person, and it's once again much more limited than tapping into the actual native activity that tells you everything about the person's intention, which we're hoping to do eventually.
Dr. Anderson?
Yeah, I agree. And yeah, this has been some debate related to using EEG and for BCIs, and I think it has been argued that it can almost do as well as some of the performance that was shown early on in the invasive techniques, but since then, clearly the invasive techniques do better. An author was talking earlier today, and we often get this comment too that why not use eye movements and closing your eyes or something as a click of a mouse. And that's true, that would work very well. And but again, I think it limits the number of, or gut movements, the numbers of degrees of freedom that you can actually have. So looking into the future, like if we want to decode a large number of grasp, or if, say, in someone that's mute, if you wanted to implant Broca's area, which is going to be much more complicated, I would think, than coding a limb movement. I don't think this would work so well. Also, but I don't know if it's applicable to many patients, maybe some locked-in patients, but we have done these decoding experiments in eye movement areas now, where the monkey just plans an eye movement, or others would say attends to a location, and we control the cursor. So the monkey actually makes no movements at all, but just generates a planned eye movement. So you could imagine someone is completely locked in. If you implanted an area like the area LIP or frontal eye field, you could help them in that regard.
Great. I think it's, I think the kind of technologies that will be required for non-invasive monitoring, and certainly for non-invasive manipulation of neuronal function are technologies that we haven't imagined, because I think the problem is because it's so difficult that it's going to take technologies that are really going to surprise us. So I think it's a long way off before the kinds of controls, the kinds of specificity that are required for neuroprostheses are going to exist in a non-invasive form. And I think we just need to, I just try to accept that. And I don't think about invasive approaches and non-invasive approaches at the same time. I think they're going to stagger in time to quite a surprising extent. And I think it's just going to be a long time before we see that. And I think that once we start doing more and more invasive approaches, more and more invasive neuroprostheses, that we'll become more accustomed to it, and the kind of barrier that exists to opening up the head won't be as strong, and we'll start to think of invasive neuroprostheses the same way we think about cardiac control, and the way we think about look at deep brain stimulation. There's something that we do quite regularly now. So I think that there'll be more and more acceptance of invasive approaches to control, to monitor the brain, control the brain, and I think that we'll just have to wait for whatever the novel technologies are going to emerge that will allow us to do the same thing in a non-invasive form. So.
Thank you, Dr. Berger. So, Dr. Anderson, just following on what Dr. Berger just spoke about in terms of new technologies potentially coming down the pike that might enable some of these things. And I know that by the time technologies are packaged in a way that we can actually see it and hold it, right? It's pretty far down the line of development. Can you make some comments on, if you could look into the crystal ball, of what types of technologies might emerge to greatly change the landscape for this BCI?
And Richard's got a Rich has got a ball too.
Yeah, right.
He hides it, but I know he has one.
Well, I think things that I've heard people talk about, they're all going to cost hundreds of millions of dollars. They may and so I guess that's why I even mentioned the brain mapping project, because some of those technologies may apply there, but there are things like molecular approaches that may be able to label cells that are active at different times and at the cellular level. Another is maybe little beads that you could inject into the brain that could record signals and transmit them wirelessly, so you could fill up area of the brain. There's some people working on imaging techniques. You can do things like laser pumping to, right now I think they use xenon to get really strong MR signals, and so then you can use very weak magnets and get enormous signals, but there are problems with that, associated with that as well. So there are things you can imagine, but some will fail, and price tags probably on them would be, so I don't see anything really immediate other than incremental things that we can do now in the immediate short term for labs like myself, but there are big ideas out there.
Great. Dr. J, any further thoughts on that?
Yeah, just to add to that list, there's also people working on the carbon nanotubes or something like that would sort of integrate themselves into the brain tissue and move with it, so you don't have the problem of them scratching up the brain as things move. All these things have been under development for a while, but I haven't seen like tremendous progress along any of them. I think, as far as I know, the black rock array is still the state of the art. Just using more of those, miniaturizing them more, I think will be very helpful. We're already getting 7 degrees of freedom of control pretty well with just 2 arrays. We could go a lot further with that technology.
Great. I'd just like to wrap up in the next few minutes, but one thing that we're, Dr. Peck and I, Dr. A's and all of us here, we're impatient in a way, right? We like to see, we take care of patients. We like to see patients get better, and we like to see these sort of technologies move through the sort of research and scientific realm and emerge so that we can begin to embrace them and understand how to integrate them into our activities to take care of patients, whether it's on the sort of the trials side, that such as the projects that we've been talking about. But what do you see as the key bottlenecks for these technologies to make it from the laboratory to our setting? And I'd like to let Dr. Berger take that question first.
Well, I'd say 2 things. One is the mapping, so to speak, the convergence of medical needs and what I'll call experimental needs. I'll mention that again. Is that you really can't deploy the kinds of prostheses that we're thinking about until you can also meet a medical need of the patient. And so the convergence between those 2 sets of requirements, those 2 sets of needs, I think is the kind of thing that really has to happen. And it's clear, from the work that my 2 colleagues to the left have shown you that they've been able to do that. And that's why there's been the kind of progress that there's been, and the tremendous advances that they've made and been able to make on the technical arena, on the experimental arena, they can pass over to the clinical arena. And so when it works, it works. And you can two fabulous examples of reaching that kind of goal. So I think that that's clearly a roadblock. There are some things that, some types of prostheses that are in development or that are being thought of, and you could really see how to apply them experimentally, but not be able to move them clinically until there's this kind of convergence I'm talking about. And the other roadblock, I'll at the risk of beating the almost dying horse, I'll say again, is this our lack of understanding of neural coding. We don't understand very well at all what it is that firing rate or temporal patterns, what kind of codes neurons are using for representing what's either an internal event or an external event. And until we understand that coding process better, we're not going to be able to make or to include in the prosthetic systems we're developing the richness of information that patients and that you and I have in our daily life. It's just, I mean, it's so obvious. I feel kind of silly stating it, but there's a real need to understand more about how neurons code information and how events are joined together internally in the brain. And without knowing that language, without understanding that code, there's only so far you can go, and that's not very far in terms of being able to understand neural behavior and thought processes, etc.
Dr. Anderson?
Yeah, so I think, at least for the motor prostheses, which is similar to other medical devices, there's a big gap between demonstrating, even in phase 1 clinical trials, that they're safe and effective, and actually getting it to the clinic. That's quite a hurdle, and it takes quite a bit of time. So I think that may be a major roadblock there, although certainly all of us want to see this in the clinic, otherwise we really would be working on, well, I mean, scientifically interesting, but the eventual goal is to, or earlier goal hopefully, is to get it in to general use.
Dr. J?
I want to echo the sentiments of Dr. Berger and Anderson. I think understanding the neural code will be very important. Just keeping on, pressing on, trying to understand as much as we can. As we know, for example, that cosine tuning isn't the end of the story. For motor cortex, it's extremely noisy if you treat them as just cosine tuning curves. There's a lot of dynamics. I'm sure there's a lot more information we could be tapping into there. And the issue of whether this will ever be, that motor neuroprosthetics will ever be commercially viable, I haven't really figured out the answer to that question. Different people I talk to think different things about it, but maybe there's some way to bypass that sort of typical way of getting things to the clinic. It's going to be a very small population that we're helping. It's never really going to be profitable for anyone. Really, the only reason to do it is to help people with disabilities. Maybe getting funding from alternate sources instead of commercial sources will be the answer to that. I'm happy to hear other people's ideas on that.
So before I ask for final comments from the three panelists, I think it's appropriate now for me to sort of make a couple of comments that very much is appropriate. I think that following the comments of the three panelists, I think for us, when we all know that the science is absolutely fantastic. We also want to see this be relevant to our patient population. And last night, I sort of described a vision that Dr. Raisin and I, over the course of the last couple of years, have tried to create, which is that we like to make patients with disabilities better. And being an engineer and being one of many engineers in the room, we really firmly believe that technology is going to be the solution to many of these things. And so, if that is the case, how do we facilitate the transfer of this technology from the laboratory to the clinical setting? And so, we thinking about how this is done. I think that up here we have 3 world experts that represent concepts and platforms and ideas that pose 2 very different challenges for us here on the clinical side in terms of facilitating this process. The first would be, having a population of patients that we can recruit from that have a catastrophic injury, that have a huge amount to gain, and possibly, when you talk about brain and surgery together, an implantation of some electrode into the brain, you think of that as having some potential huge risk, but all the neurosurgeons in the room will tell you that that is something we do routinely, and the actual risk associated with implanting an electrode. And I joke with my colleagues all the time that when I'm not capable of doing surgery, I might actually sign up for one of your prosthetics and see what happens, because I know that the risk isn't that great, right? I mean, this is stuff that we do all the time. And so for platforms such as the motor prosthetics, what we really need is a very a trusted environment from which to recruit patients, and I think that the collaboration that we've established with Dr. Anderson moving forward with APL and Kent and Colonel Ling is a good example of that, right? We were very quickly able to recruit patients in that regard. But then the second type of project is one that is potentially represented by the REMIND program, which is a program, a project that requires invasive placement of electrode arrays into the hippocampus. And we're talking about patients who have relatively milder impairments, right? Patients with primary hippocampal dysfunction, or patients with traumatic brain injury, or patients who suffered transient ischemia where you see one hippocampal neurons die. So how do you even begin to integrate some of these platforms? You can't even begin to do that unless you concurrently, I think, unless you concurrently identify, as Dr. Berger mentioned, a parallel set of developments that is aimed at treating a disease process that you would do anyways, that doesn't convey any additional risk to these patients, and in parallel develop these technologies. And so we here at Rancho are very, very much aware of what these challenges are. And one of the principal visions of this living laboratory that we refer to is to provide a possibility to move forward quickly with each one of these 2 sets of challenges faced by these 2 very different problems. So I'd like to just ask Dr. J, starting with you, to make some final comments, and then we'll take some questions from the audience.
Thank you. Once again, I'm just very impressed with what I've seen today. I hadn't even heard of Rancho until I was invited to come talk here, but it turns out it's a world-class program, and I'm just really honored to be here and meet you all, and I've learned a lot.
Dr. Anderson.
Yeah, I enjoyed the conference very much and learned a lot from the session in the first half, and enjoyed the conference in general and the discussions we're having now. And I look forward very much to working with Rancho and USC and APL and DARPA as we move forward.
Thank you. Dr. Berger?
Yeah, I guess I'd like to say a couple things. One is that it's difficult to appreciate what key people can do, and the landscape of neuroprostheses is completely different today than it was 7 or 8 years ago because of Jeff Ling. I'm glad he's not here, so I won't be accused of trying to do whatever I was trying to do. But Jeff just came in like a windstorm and changed everything at DARPA with respect to the biological component of projects, but particularly with respect to how to look at neuroprostheses. And it's just and his influence has gone way beyond DARPA. And I'm obviously thankful for your contributions as well, Kent. But I've worked mostly with Jeff, and we were there with him when he first got there. But it's just, you just can't say enough about what it's a real lesson in how one person can just change the field, and it gives you a lot of hope and it spurs you on to be more aggressive in a good way about the way you think things should be, and when you say, oh, it's always been like that, it just can't be changed. Nope, that's not true. Lesson in point is Jeff Ling. So I'm really thankful to him, and more what I'm trying to emphasize is the lesson that he gave with his performance and the way in which he changed things. He's really quite a remarkable person, and it's a very remarkable lesson. Whatever the second thing was, I've forgotten it, and it kind of pales in comparison anyway. So I'll just leave it at that.
Great, Dr. Aiden.
You need that hippocampus clearly. I just would disagree, Dee, that this doesn't have commercial potential, because if we do look at the rate of improvement over the past few years, it's extraordinary. And the degree to which deep brain stimulation has been accepted. I mean, my best friend from high school now is disabled from Parkinson's, and she just got married last week to the love of her life, and she's moving very nicely. And I think that we're just scratching the surface. There are so many people that I've dealt with cerebral palsy who are locked in and can barely communicate. They're very, I suspect, very active minds. And I think most of us in this room are the temporarily able-bodied. We're going to live a lot longer than our parents and grandparents. We're going to get degenerative conditions, ALS. This can liberate them. Parkinson's disease, if they do brain stimulation and other pharmaceutical solutions don't work all that well, this can liberate them. On and on and on and on. We've seen some of these diagnoses. I've seen people with MS completely confined to bed with a very active mind. So I think that as the technology gets better, as the neurosurgeons become more and more known for their competency in doing this, it becomes less of this extraordinary thing to have a brain procedure. This is going to be much more widely accepted, and I think the robotics themselves are going to get better and better. And to, like Kathleen and we were really with people's spinal injury all the time. See, it's so difficult to be tied 24/7 to another person who may not even be someone you particularly know or like. You're dependent on them to scratch your eye or to take a sip of water. Just that tiny bit of self-sufficiency can make such a difference to them. So I've got high hopes for it. I mean, I've not been a trading rate, but I think that there really are. It's not necessarily something that can be self-sustaining financially.
Dr. Heck?
I was thinking just the same thing, because I think that there are probably applications in stroke even, and we know that there are plenty of patients out there with stroke. And as we move forward, we're going to figure out ways to rehabilitate people with these kinds of techniques, with all sorts of neurologic conditions. But I had a question for Ken, because there's a lot of this talk about brain mapping initiative, and we all have our own opinions about what that means. And with the experience of the Genome Initiative, that at least that approach doesn't really apply, I think, in most of our minds in terms of science and neuroscience. How do you and/or Colonel Ling interpret this idea of mapping the human brain?
So I would not want to speak for him in that regard. I will say what he said to some of his colleagues at DARPA, but first it should be understood that this is, we're talking about what the president said in the State of the Union, right? He's going to give $1 billion, I think it is, over the next, or 10 million, I don't know, some billions of dollars over the next 10 years to map the brain. But the operative word in that is dynamic mapping of the brain. So that's a little bit different than just understanding where the different landmarks are in the brain. So it's a dynamic mapping of the brain. Now, my understanding is it's primarily an NIH and NSF initiative. And when Dr. Ling was talking to his colleagues that are kind of bio type of people in the office at the Defense Sciences Office within DARPA, his thoughts were that DARPA should have some sort of role in that. And we've got one of his colleagues, Dr. Matt Goodman, is working on advanced brain imaging. So he's probably going to try to get that lashed up to the program somehow. Another colleague, Dr. Bill Koblenz, is working on the MEG initiative in Princeton. He just got kicked off up at Princeton for some MEG effort there. So, so Dr. Ling wants them to link that to the brain mapping project to some extent, and he's looking for other ways to assist in that as well. So you should also understand that Dr. Ling doesn't sit still very much. He has a lot of things going on. And when he first started as a DARPA program manager, he wasn't even employed by DARPA. He was an Army colonel working at USIS in the Department of Neuroscience, and doing neurocritical care in Johns Hopkins University's in Johns Hopkins Hospital. So he wore many hats. Well, he evolved over those 5 years, and he retired last August from the Army, lost his affiliate, or let go of his affiliation with the Uniformed Services University of Health Sciences, and he acquired last year an opportunity to work at the Office of Science, Technology, and Policy. So they're kind of the ones that are driving this dynamic mapping of the brain. And so in his capacity as working at OSTP, which is in the executive office, so working right with the president's staff, he has other opportunities to reach out to different types of people that DARPA can reach out to bring them into the community of the advanced dynamic brain mapping. So that's why his reach is much farther than I can address, because he's got this whole other life that he lives at OSTP, and he brings people in through that avenue as well. So he's interested in having DARPA play in that brain mapping. In fact, he's already doing some brain map repair. Essentially, is trying to map certain parts of the brain right now. That's one of his initiatives. So if he can feed that information into the effort, then he will. It's all about advancing the science and advancing the ability to help people. So if that addresses what you're looking for, Dr.
Yes, thank you.
Other questions, comments? Dr. Azen gets the last word.
No, the last word is everybody's invited for dinner across the hall to continue the